Since early satellite launches in the 1960s, numerous bacterial experiments have taken place in space. The results of these experiments have revealed something very strange. Bacteria, particularly non-motile, suspension-grown organisms, seem to behave differently in space. They exhibit abnormal growth, including a thicker cell wall. They reach higher cell counts and produce secondary metabolites with higher than expected frequency. These altered physical characteristics are accompanied by increased capability to cause disease, improved biofilm formation, and a reduced sensitivity to antibiotics. These changes pose a threat to human health, particularly as every human who travels into space unwittingly carries with them an ecosystem of microorganisms on their bodies, and the internal surfaces of the ISS are teeming with diverse populations of bacteria. A model exists which theorizes that these changes are related to a reduction in the transport of molecules in the environment around cells. On Earth, gravity creates currents and turbulence, which help molecules move down their concentration gradients. In space, however, there is no gravity, and so molecules can only move around by random Brownian motion, a much slower way to get around. This model can be tested by looking at the genes expressed by organisms in spaceflight, as these would reflect the environment around the cells. If transport to and away from the cell is perturbed, nutrients would not reach the cell consistently, and it would express genes associated with nutrient starvation. Reciprocally, secreted waste would build up around the cell and elicit systemic responses to toxicity. The aim of this investigation was to assess the reduced transport model by investigating the local environment surrounding cells in microgravity. The investigation was run at both 0G on the ISS and 1G on the ground. It took place in specialized test tubes with 4 chambers containing sterile growth medium with a glucose carbon source, the bacterial inoculum, antibiotic, and a fixative. Pushing right to left would move the septums separating the chambers into the bypass, allowing the solutions to mix, and then ultimately fixing the cells for analysis on return to Earth. Gene expression was evaluated via RNA sequencing, as transcribed RNA indicates which genes were activated by the microgravity environment. Those cells were exposed to 3 different antibiotic levels. The effect of gravity was the sole focus of this investigation. A 4th sample, with no antibiotic, was also included, but unfortunately it was terminated prematurely. The antibiotic data collected from this investigation has been published elsewhere. Gene expression data indicated that cells in space were under starvation conditions and exposed to both high levels of acetate as well as acidic pH. This scatterplot depicts the relative expression of genes at 0G in space relative to their 1G controls on Earth. Based on RNA sequence data, genes whose expression in space differed by at least 2-fold were considered differentially expressed. The points in red correspond with genes overexpressed in space relative to Earth, blue represents non-differential expression, and green represents genes that were underexpressed in space. The pink triangles, white circles, yellow diamonds, and purple squares are genes related to metabolism, low pH, and high acetate levels, and were all significantly elevated in space. Starvation conditions were suggested by the following observations. Despite glucose being present in the growth media, some glucose starvation-associated genes were expressed in space. For example, the DPS gene, which encodes a protein that protects DNA during starvation. Further, genes for importing and using alternate energy sources were also overexpressed in space. This includes a number of maltose transporter genes, despite the fact that only glucose was supplied. This resembles the risk-prone foraging strategy seen in starving animals. The cell expends limited energy on being able to metabolize other resources, without even knowing if they're available. Genes involved in glucose metabolism were also upregulated. Most significantly, glucose catabolism to acetate. Acetate can be scavenged as an additional source of energy, but while outside the cell, its acidic form is toxic and can lower the pH. Increased metabolism also encompassed higher expression of the TRP operon. This is involved in amino acid biosynthesis and produces organic acids during this process. With this in mind, space samples unsurprisingly indicated systemic responses to acidic conditions. This included an upregulation of the glutamate-dependent acid response system, the most efficient of the 3 acid responses in E. coli. Based on this gene expression data, the altered extracellular model was proposed. In the absence of gravity, nutrients are consumed but not replaced fast enough, leading to metabolic starvation responses, including acetate production. Acetate and other acids are exported, but do not move away fast enough, lowering the pH around the cell and subsequently eliciting systemic responses to both acetate and acidity. So, how does this model fit with the weird physical characteristics that we've observed? Well, the increased metabolism that results from insufficient nutrient transport can cause higher cell counts. This is already exploited in biotechnology. Cells that are fed incrementally reach higher cell counts than those that receive a constant supply of feedstock. On the other hand, the lowered pH might be the culprit behind bacteria's apparent increased capability to cause disease in space, as numerous virulence factors are enhanced under acidic conditions. Lastly, if molecule transport is reduced, antibiotics probably aren't effectively being delivered to the cells. This could lead to the antibiotic resistance that has previously been reported. These findings have important implications for astronaut health. Humans have constantly been in space since November 2nd, 2000, surrounded by an ecosystem of weirdly behaving potential pathogens. Understanding why this behavior occurs is the first step to implementing preventatives and treatment strategies for safe space travel, something that will be increasingly more important on longer space flights, like the one planned to Mars in a few years.